Here's a study that was performed in a rare disease genetic population of individuals that had inborn errors of the mitochondria. So these are individuals that have sequence-verified mutations that are occurring in one of the 1,000 or so different genes that affect the mitochondria. These have clinical heterogeneity, and given the fact that there's many particular genes that may be altered and biochemical pathways that may be altered within the mitochondria, these individuals are extremely tough to diagnose and follow clinically. Now, when we look at over 100 individuals with sequence-verified mitochondrial disease and ask once again, are there markers that are altered in their blood relative to age, gender, demographic matched counterparts, what we find is the data here on the left. Again, we can capture thousands of circulating small molecule biomarkers across these diverse populations using our underlying RLC technologies. And when we ask, are there specific molecules that are altered in individuals that have mitochondrial disease, what you find here is in the inset, in that there's a subset of molecules that are altered specifically in individuals with mitochondrial disease. Now, once again, as was the case with the NASH example I just showed you, all of these markers represent unknown sort of small molecule biomarkers that have never been classified. When we look at their underlying structural relationships, we find that over a dozen of these markers, as shown here on the red dots on the left inset, all are chemically very related to one another and represent a whole new biochemical pathway that hasn't been described before in humans. Now, all of these markers are once again altered in individuals with underlying mitochondrial genetic diseases. We can isolate out one of these that's shown as a blue circle here, solve its underlying structure, develop targeted assays that allow us to technically replicate this data, as well as biologically validate these now, once again, in independent clinical sets. So when we look at an independent data set of patients with mitochondrial disease, what we find is control patients have very low levels of this marker. Patients that have very localized mitochondrial disease affecting only a single cell type within the eye, once again, have very low levels of this marker. And in individuals that have systemic mitochondrial disease, again, through genetic means, have quite an elevation in these underlying markers. Now, as was the case with NASH, even though these individuals may have shared genetic mutations that ultimately result in their mitochondrial disease, you'll see that there's a massive spread in the underlying levels of these markers. Now, this spread has incredibly important prognostic information and very much varies with the degree of disease severity that we observe in these individuals. Now we can begin to ask, using formal ROC analysis, what's the diagnostic sort of value of this single biomarker once again. And what we find is that in independent populations, the AUC is about 0.8 as a diagnostic test. This is something that beats current genetic testing in these individuals. Once again, that is quite complicated given the thousands or so genes that may be potentially mutated in these individuals. As is the case with the NASH molecule, this is a biomarker now that's been advanced to our high complexity CLIA laboratory for development of a CLIA diagnostic assay. Now, what we can do as part of this rare disease population is we can begin to ask, what are those genetic variants that associate with the circulating marker, even in normal individuals? And when we do so, you develop the Manhattan plot that's shown on the bottom right. Once again, we can localize the enzymes that produce this underlying biomarker, as well as the transport systems that allow them to be transported into the cytoplasmic space, from the cytoplasmic space into the blood. And when we do so, again, we can localize this to the mitochondria. Now, this is a marker that we've been able to validate in vitro and cellular systems, both in iPS-derived models of mitochondrial disease, as well as in fibroblasts isolated from human patients. This is also a marker now that we've validated in preclinical models across a number of transgenic animals that have mutations within the mitochondria. Now, as often the case with our rare disease populations that we study here at Sapient, we're very interested in understanding how these rare disease populations may also map onto common human diseases. Now, as you're well aware, the mitochondria has been implicated in a host of human diseases and biological processes, everything from heart disease to neurodegeneration and Parkinson's, as well as even biological aging. Now, the challenge with studying the mitochondria in vivo in humans is that there's no good measurement that allows us to interrogate mitochondrial function in vivo in humans. And this requires either isolating out tissues and studying them in very artificial systems, or measuring very indirect metrics that provide very poor insight into underlying mitochondrial function. In our human biological database, however, we can begin to ask, how does this one singular biomarker that we use as a readout for mitochondrial dysfunction, how does this map onto various diseases and biological processes, including age? And what we find is that across over 100,000 diverse individuals from around the world, there's a very, very consistent pattern whereby somewhere in adult age, around age 48, 49, this is a marker that begins to elevate in blood and linearly increases from about age 50 to approximately age 100. And this is some of the first real world evidence in our hands to suggest that there is progressive mitochondrial dysfunction that occurs with human biological aging in vivo in individuals. Now, if we pick any particular chronologic age here, whether it be 70 or 80, and ask individuals that have higher levels of the marker indicative of greater mitochondrial dysfunction relative to individuals that have lower levels of this marker, are they prone to certain diseases? What we find, in fact, is that having an elevation at any given chronologic age, indicative of more accelerated mitochondrial aging, is very much associated with certain disease processes around cardiometabolic disease, as well as age-related tissue degeneration. Now, as mentioned, this is a marker that's been advanced to our high complexity CLIA laboratory for development of a diagnostic test that will ultimately be used in humans for rare diseases. But also, this is a marker now that may be leveraged as part of aging and longevity studies, as well as for the development of therapeutics in the cardiometabolic and other tissue degeneration spaces that are targeting the mitochondria.